Okay, so it looks like we have a number of researchers, administrators, and faculty on the call, one student, and looks like all groups are represented. So we're again thankful for you guys to join us this morning. We're in for a wonderful presentation, and I would like to get started. So welcome everyone to the National Academy of Inventors ScholarShare webinar series. My name is Jade Stewart, and I currently serve as the Director of the National Academy of Inventors. For those who may not be familiar, the National Academy of Inventors is a member-based organization comprising U.S. and international universities, government agencies, and nonprofit research institutes with over 4,000 individual inventor members, fellows, and senior members spanning more than 250 institutions worldwide. The NAI was founded in 2010 to recognize and encourage inventors with patents issued from the U.S. Patent and Trademark Office, enhance the visibility of academic technology and innovation, encourage the disclosure of intellectual property, educate and mentor innovative students, and translate the inventions of its members to benefit society. We offer a variety of programs for our members and partners, including but not limited to our mentorship platform called Global Academic Inventors Network, commonly referred to as GAIN, which connects students, researchers, and faculty from across the world. Our multidisciplinary journal, published in-house, Technology and Innovation. From Campus to Commerce, our video series featuring early stage innovation that takes place at our member institutions, such as the inventors of plant-based protein alternative meat, Beyond Meat from the University of Missouri. We also offer 2 recognition and award programs, fellows and senior members. We are still accepting nominations and encourage you to submit an application or nominate your peers for these awards. Our organization is committed to using its platform to help minimize barriers and promote an inclusive environment for all, especially through these programs, as I mentioned, and beyond. Though we were not able to have our annual meeting this year, we are excited to present our virtual ScholarShare series. Today's webinar is titled Microscopy-Aided Design and Manufacture, MADAM, as a Seed for Innovative Ecosystems. And I'd like to introduce our speaker. Melissa Kanota-Tate is the inaugural Paul Traynor Chair and Professor of Biomedical Engineering at University of New South Wales. She is a global thought leader in healthcare innovation. Her R&D program is epitomized by its cross-cutting nature, where the fundamental laws of mechanics are applied to biological medical systems to elucidate and emulate emergent and smart properties. Melissa is the recipient of more than 40 honors and awards, including Engineers Australia Most Innovative Engineers and the Christopher Columbus Foundation, U.S. Chamber of Commerce Chairman's Distinguished Life Sciences Award. She holds a number of patents and number of foreign patents that have been licensed to 3 companies. She's the founder of a consulting company and 3 startups, and she has published over 300 articles in peer-reviewed conference proceedings and serves on the editorial boards of journals such as Current Opinion and Biomedical Engineering. Dr. Tate is also a fellow of NAI, so please join me in welcoming Dr. Tate.
Thanks very much for the introduction, Jade. Thanks, Jody. I also thank the NAI for this great opportunity. Hi, everyone out there. I'm sorry I can't see you directly or we can't be together directly, but this is great that we can use technology to hopefully make the world a better place and deal with challenges that we're all dealing with as a global society and as a community together. At the start of my presentation, I'd like to acknowledge country. I acknowledge the traditional custodians of the lands on which I work and live, and I recognize their continuing connection to the land, water, and community. I pay respect to elders past, present, and emerging. And it's quite interesting to know that the Aboriginal Australians have been the custodians of the land on which I'm working and living for the past 60 to 120,000 years, to put history in context. I also have a few disclosures, as Jade indicated. I've been very involved with commercialization of technology, right from intellectual property protection through patents, as well as licensing to companies and starting up companies. My current startups are all in a pre-revenues phase. Today, we're going to talk about a topic that's very near and dear to my heart as far as innovation goes and invention. I'll start with an introduction to the topic and then talk about 2 case studies that hopefully will make the technology come alive for you, and also the idea of innovation and learning from nature come alive for you. And I'll conclude with the way that we're using our technology platform as a seed for innovative ecosystems. When you see these 3 trees, insect joints, and animal joints in front of you, you may not think that they have much in common, but they have remarkable smart properties. In particular, their mechanical properties are spread in space and time in such a way that this gum tree or eucalyptus tree, which is almost as long as a football field, bends like a blade of grass in a gust of wind. This grasshopper uses the distribution of mechanical properties that go seamless across its leg joint to propel itself across fantastic distances with respect to, compared to its own body length. And of course, our own joints exhibit remarkable smart properties as well. The gum tree here, I'll just show this is taken just on my iPhone, but shows this is a gum tree that's probably nearly 100 meters tall. You can hear the wind, you see it bending like a blade of grass. As it bends, not only is it not snapping and failing in the wind, but it's also propelling the fluid within the tree from the roots to the tips of the leaves, which is quite remarkable. In that way, it keeps the tissue of the tree alive. There are remarkable parallels in ecosystems which might seem as different as the Amazon River basin and the human body. What's very interesting, if we approach these different ecosystems as biosystems with different spatial and different temporal scales, we can start to better understand the world around us and use this understanding to develop new technologies that can solve challenges and problems to make the world a better place for everyone and everything. These length scales run from nanometers to kilometers, from seconds to centuries. Centuries if you're thinking of how long it takes this forest to form, from the formation of the first tree to the formation of the full forest, and now the unfortunate deforestation that is affecting the health of the forest and the continent. The spatiotemporal scaling is perhaps for me most impressive when I think back to a vertebrate biology class I took as an undergraduate, where I learned that in utero we go from 2 to 70 trillion cells from the event of fertilization to becoming a full-grown adult with 70 trillion cells inhabiting our bodies. When we think about evolution and the concept of every single child that goes from the moment of conception through to growing up and going through life, goes through this process of vertebrate evolution that is evident also throughout evolution of vertebrate species. And this incredible capacity of these biological systems to develop and evolve, that they build their own materials, and that material adapts to the dynamic environment in which the material, in this case, the cells themselves reside, is quite an impressive feat. And if we could understand how those brainless cells do this, we can then use this information to create smart technologies and to create more sustainable materials. We talk about these properties in terms of emergence, and try to understand them in a way to recapitulate them in the way that what we say postnatal healing for those after birth in the postnatal period it recapitulates or it repeats these developmental processes throughout life. And we can talk about this more in the discussion if you're interested in the concept. But it's reiterated very much in invertebrate biology books. The cells within these systems, whether it be an embryo or whether it be a tree, they respond to mechanical signals in their environment, these dynamic signals. In this case, they're plotted here on a log plot of dilatational or volume changing stresses versus deviatoric or shape changing stresses, and versus time here. And you can start to see how these the physical environment and the dynamics of the physical environment literally shapes the cells and directs the cell fate to become the different tissues and to generate the different tissues of the body. And so here's a stem cell, some early work from my laboratory showing that in response to very small mechanical signals, these cells start to create in the nucleus, they create their own structural proteins, such as collagen here in green, which then gets excreted to the extracellular matrix and forms the tissue over time. So you can see this happening over time and over different length scales, from the cell itself at 5 microns here, length scale. This is the tissue itself forming over time, as indicated with the colors here, chelating, bone forming within a defect via stem cells over time. And here's the whole bone defect that itself heals over time in response to mechanical and biochemical cues. In the first case study, I'm going to talk about how we came up with a new technology to develop textiles that emulate the smart properties of our own tissues. The concept started quite a ways back. This is some work from one of my closest clinical collaborators, who's also my partner in life, Dr. Ulf Knote. And this is an early work of his with Dr. Dempsey Springfield in New York, in which they did a one-stage bone transport. This is a young patient, an 11-year-old male, where they resected out the fibula and left a tissue called the periosteum in place. You can see the periosteum here on the left in A. They took the fibula and moved it to the tibia. So you can see here a tumor was removed, the fibula was moved to as a graft where the tumor was removed. And you can see over time how the bone regrows within the periosteum, which is like a soft sock-like sheath. And the massive bone graft that was moved over integrates over time. In trying to understand this remarkable feat whereby these cells can regenerate so much tissue in a relatively short period of time, and they do it so well, we've carried out a whole series of studies. And Dr. Knote has also studied this in adults where they need more mechanical support through these implants. In this case, this is a patient with a dysplastic hip where the joint needed to be replaced and there was an uneven leg length left to right, and he corrected that using a similar procedure. And similarly, these stem cells can grow the tissue remarkably large volumes and in short periods of time. We tried to understand how the cells do this, and we also, in so doing, tried to understand this remarkable tissue called the periosteum. As I mentioned, it's like a hyperelastic sheath or sock that covers all the bones of the body, and it's velcroed literally with collagen fibrils to every bone of the body except surfaces that are covered in cartilage. And so this is some work where we did high resolution, what we call digital image correlation, which is a method by which we can understand the strain environment of the cells within the periosteum at a very small spatial resolution. It's like looking under a microscope, but in the lab, not under the microscope, but looking within the environment of the cells. And in this remarkable video here, you can see the color map here as it changes over time. We're loading this bone to mimic stance shift of a person or an animal after getting surgery. So they're shifting their stance back and forth, which causes very subtle changes in the mechanical environment of the cells within this hyperelastic sheath called the periosteum. And we were shocked when we saw this because we assumed this would be a homogeneous strain field. And we saw that it's really very heterogeneous. And we wanted to understand how the tissue has these properties and how it transmits its information to the cells which build the bone within the defect behind this periosteum. This is some work where we did some high resolution imaging of these cells to understand the local environment at the length scale of individual collagen fibrils. And here you can see these collagen fibrils are like curls of hair. And when they're tacked down to the bone like Velcro, the curls are stretched out. And when there's a trauma to the bone, the curls relax. And in so doing, they change the conformation of the cell, which we believe instructs the cell to start building tissue. So these were key findings. This is just some data here showing this is with the fibrils tacked onto the bone, and this is without, and you can see the degree of crimp in the collagen fibril itself and how that's transmitted to the cell nucleus. So it's really like curls of hair, but at much, much smaller length scales. So we went on to try to understand these properties of periosteum using digital image correlation to understand the strain fields in here swatches of periosteum, and here doing some mechanical testing and seeing there's this this strain stiffening, so the periosteum actually becomes like a plaster of Paris or a cast around the bone under impact loads. And has very different mechanical properties under regular loads, just with walking, for instance. So we characterized the tissue's mechanical properties, and we also then were very interested in characterizing its other advanced properties, including its permeability. And here you can just see the pre-stress in those collagen fibrils, whether it's present or not, basically directs the direction of the permeability and the degree of permeability, where B and M are bone to muscle and M to B is muscle to bone. And here you can see it also very much directs the volumetric flow rate. So this tissue has a number of smart or advanced properties that would be worthy of emulating. This just shows the system in situ as it would look if you could cut through a bone or through a joint. And here you see the muscle, and here you see the bone, and the periosteum is here. It's literally a membrane, a smart membrane that regulates flow based on stress state. In the follow-on study then, we wanted to really understand at that molecular level how the periosteum is organized. And so when we're traversing length scales like this, again, in this case, we go to high resolution microscopy, and we started then to map using, there's a lot of detail with second harmonic imaging and dual photon and multiphoton imaging, where we can map then the elastin fibrils within the periosteum and the collagen fibrils. And here you see this looks like a beautiful weave in 3 dimensions. And we can start to understand explicitly what is the design parameter underpinning this tissue and its remarkable properties. So this actually inspired me to think, okay, well, how does it come to be that way? And how can I create a textile like this in the laboratory or in my prototyping workshop? So I had this idea, I had this strain map of the tissue, and I thought, well, what if instead of mapping colors, if I was wearing a colorful blouse, for instance, with a pattern of flowers or whatever pattern, what if I mapped these patterns of strains or mechanical properties of the fibers making up the textile that's the tissue? So in computer science, the basis of all algorithms is recursive logic. So I applied recursive logic to the problem, and I thought about if I could, in theory, unravel the tissue, what would the thread look like in terms of mechanical properties? And then I came up with algorithms where I could, this is a single solution for this particular tissue, but there are, of course, millions of solutions, and I came up with algorithms to optimize those solutions to actually create textiles in the lab that emulate the properties of this remarkable tissue. And that became then the basis of, that's intellectual property, which we've gone on to protect as a family of patents that are, some are granted and some are pending. And this just goes through the process where we image and render the distributions, import them into a computer-aided design program. We scale up and create motifs for the textile patterns. We import them into weaving programs and manufacture thereby novel, mechanically responsive textiles using a computer-controlled jacquard loom. And then once we have these textile prototypes, we test them for both internal and external applications, which I'll show you in a minute. So this just basically shows the process. More recently, we've actually integrated the process into other advanced manufacturing technologies, including 3D printing and other rapid prototyping modalities. And this just shows here some examples of different patterns that we can tune to exhibit different properties. It shows the mechanical consistency of these properties and basically demonstrates the feasibility of creating textiles that emulate the PERIOSTEUM. Here are just some examples of different textile swatches created and tested in that way. Just to bring the concept to the highest level, then we tune the patterns, we tune the materials for target stiffnesses. So here you can see with elastane and nylon, E and N, and S is silicone. You can see the different combinations. If you're interested in the full details, these are published in the publication noted here. And in this case, if the target was to achieve bulk mechanical properties similar to PERIOSTEUM, we would be in this range. If you go to the next step, then, so we're in the range I just showed. And then if we're thinking about implanting these, we also have to think further down with the process that we're also going to need to sterilize them. And here then we see the effects of sterilization, different types of sterilization. And again, just comparing to PERIOSTEUM, if that were our target tissue, you could use this modality, this design modality and manufacturing modality for any possible tissue, including tree bark, if you wanted to. This just shows the further development of the technology to incorporate gradients, for instance, gradients in here in this longitudinal direction or in the radial direction, for instance, and the manufacturing and testing of those. Here you can see again, we can then really achieve very different mechanical properties depending on whether we're incorporating gradients here with the grad or whether we're using a more traditional weaving pattern. So we're applying this to both wearables, which would be external applications, of course, and to implantable applications. And here you can see on the left an example of a compression sleeve, for instance, that has a mechanical gradient in the distal to proximal direction to promote flow back to the heart in the case of swelling or edema. And on the right here, you can see an internal application, for example, a membrane for a bone defect. This can also be applied to surgical meshes and stents. Just again, some images showing potential wearable applications and compression garments using the same type of mechanical engineering approach to assess structure and function. Just to give an idea from now that we've brought it then from intellectual property creation and protection and then to product development, assessing the markets. For instance, in the case of surgical meshes, there are many different applications that are listed here that would be applicable for the technology. The scope of the market is huge. There's a real need right now for better surgical meshes. There have been a number of surgical meshes that have been recalled from the market because they don't adequately replicate the properties of native tissues, and that's an advantage of our particular technology. Disadvantages of current surgical meshes that don't incorporate tissue similar properties are that the stiffnesses don't match and they become embrittled and they may shorten, and there can be real issues with these. All right, just a second case study I wanted to share with you to show a different application using a similar approach, the MADAM approach, microscopy-assisted design and manufacture. The second case study relates to flow directing materials. It was actually, the second case study actually occurred before the first case study, but they actually flow better, literally, in this order. So again, this relates to engineering of advanced materials and scale traversing methods and applications. So these are paradigms that are applicable throughout our approaches and our needs for engineering better solutions. I always emphasize multi-scale properties really do matter, and sometimes when we can't really understand or visualize how properties emerge from the basic parts, computational modeling coupled with imaging is a really powerful method to start to understand and decipher these advanced properties, these smart properties. So this is a case, this is a cross-section of bone here on the bottom left, and you can see this bone, this is a long bone, I think it's a ulna. And this bone has a marrow cavity in the middle. There are a number of, there's basically at least 2 calibers of porosity outside of the medullary cavity, including the blood vessels. And then so the blood vessels look like this. That's the vascular porosity. You can see them, the big holes lighting up with fluorescent tracer here. And then there's what's called the lacunar porosity. And those are the pores that are around the cells themselves. The cells sit, the osteocytes sit in these lacunae, or these holes. The image, we use imaging techniques then and segmentation techniques to separate these 2 calibers of porosity. And if we do a heat map here where warm colors indicate high density of pores and cool colors indicate low density of pores, you can see the remarkable difference in patterns of densities for these 2 different pores. And if we implement those and put them into bone models to better understand the flow through the bone, and I should also mention, for those of you who don't know, our bones aren't just brittle like chalk. They're actually, not only are they living ecosystems, but they're more like coral. They're full of fluid, and that fluid bathes the living inhabitants of the bone, which are the cells. And it's the cells that remodel the bone over time and help the bone adapt over time. So if we input these specific patterns of porosity into our bone models, we get flow in a direction that's counterintuitive. Well, what does that mean? If you have a kitchen sponge that's completely full of fluid and you push on it, the fluid usually goes out of the sponge. In these models, we started to observe that in certain areas of the bone, when we pushed on it, it actually imbibed, or the fluid went into those areas rather than being pushed out. And so we called that counterintuitive flow. And we did a lot of computational modeling to better understand how this could come about. At first we thought, oh, it's a bug in the program, but after a certain amount of time going through programs, you realize, no, this could be a real biophysical effect. And we use these computational models and high resolution imaging to elucidate how this was coming about. So this just shows, these are random fields of view that were captured to create these high resolution porosity maps. And then what we did was we built very simple models of pores and materials like bone. And this is a simple model. It's a modular model. So all this means is this could be repeated 1,000, 1,000,000 times to replicate any structure that has porosity. And this particular structure, I compare it to a key on your computer keyboard. So if you're to press on this key from the top with your finger, this key has different properties in these dots. they have different properties than in the surrounding areas. And here we map, this is a computational fluid dynamics model where we map the flow rate in meters per second. Here it's coming out of the plane or out of the surface of the key in pink and red, and in blue and green, it's being sucked into the surface, the fluid. So you can see these, we call these delivery dots. They're pushing, if I push on this, this porosity model with fluid in it, around these dots, we're getting a delivery of fluid, and in the surrounding areas, it's sucking in. And here you can just see in a cut through. Got everyone else's heart rate going. At least I'm not going to sleep. It's nighttime here. Sorry about that. Let me continue before we were so rudely interrupted. Let's see. So, so just to come back to this theme now, by using this combined computational model together with our understanding of how bone porosity is organized, we developed a new understanding of transport to these non-motile cells, the osteocytes that are stuck in place and depend on this flow to get their nutrients and to get rid of their waste, right? So we basically in that way discovered a flow directing mechano-active material, and we found the mechanism underpinning this remarkable property. The porosity controls the flow direction, and the permeability controls the magnitude, the flow rate in and out. And so here you can see if we flip the dot porosity and the surrounding porosity, we can have the dots as either delivery dots or imbibing dots and vice versa. So this just gives you an idea. We can completely control the system in that way, and it made us really understand a lot about bone. And there's a lot of interest in the technology as well now for flow directing materials. We have protected this internationally with a patent as well. There's a lot of interest from diaper companies, as you can imagine, as well as for drug delivery systems and other products. So mechano-active wound dressings, textiles, and body armor. So that's another nice example of how this Madame paradigm works. So in conclusion, I've talked about very different biological systems, but hopefully I've shown you examples that give you insight and help you to visualize the process of what we call Madame. The process brings us from invention to data to products. So I've talked about textiles and mechanically active wearables. And here's some examples from lingerie to athleisure wear, garments as an extension of our bodies and an interface with electronic devices and gear. There are a number of applications in sports, military transport, implants, and other fields of use. Whenever we develop the technology, we always want to make sure that we have, and particularly when we're ready then to commercialize the technology, we need to have a value proposition. And one of the most, particularly through this whole COVID pandemic, we realize how precious health is. And the value proposition of our health as a human right is incredibly important. And how do we measure that value proposition as we go to commercialize technology? And the commercialization doesn't have to be to make you a gazillionaire. The commercialization is to bring that value proposition to as many people as possible. And that may be through savings of money to make things accessible to more people, through innovation, and through access, for instance, to remote communities who aren't near a university hospital or don't have the kind of access to healthcare and products that others might have. So I won't go into great detail otherwise about this, but I think it's incredibly important for when we bring innovation to people and to improve human lives. The other thing I've described today is sort of this, I see this figure that I've created as a multidimensional aspect or representation of how we innovate. On the top, you can see this is a Qantas flight map. And although we are not flying now, we're certainly communicating via this webinar, via all sorts of technologies. And the second dimension here is crossing disciplines and the incredible importance of crossing disciplines and making discoveries at interfaces between disciplines. I have a lot of tech disciplines listed here, but I think the humanities are equally important, and the arts in this process. Then also taking a cross tissue and across organ and across biosystem view of the world is incredibly important. And when I grew up as a young scientist, as an undergraduate, we learned that you know, you focus on one tissue, you focus on one molecule, you focus on one cell type, but the importance of crossing genres, so to speak, is really, really important. And then I sort of take as the next level then, just as we can look at all these dimensions, I go from discovery to prediction to codifying those predictions into laws of biology and physiology, which there's a whole, all these laws need to be codified. We don't have them yet. And then emulating and inventing, which is an incredibly creative endeavor, and then scaling up our inventions and our innovations to disrupt the world, and disrupt in the most positive way to make life as good for as many people as possible. I'd like to conclude this talk before we start chatting together and hopefully discussing any questions you have. I'd like to acknowledge my incredible team. It's a picture of my team from 2013, before I left Cleveland to take the Paul Traynor Chair in Sydney, which was a wonderful opportunity given to me. Here's my group starting out in Sydney. And I'd like to identify individuals. This is Eric Anderson. Dr. Eric Anderson is now the chief fluid dynamicist at the National Oceanic and Atmospheric Administration. He's taken his knowledge of periosteocytic flow to understand flow in the largest freshwater system in the world, the Great Lakes. This is Min-Jae Song. Dr. Min-Jae Song has gone on to NIH. And let me just go through Dr. Sarah McBride, Sarah Evans, who did her master's degree and went on to GE Healthcare, Shannon Moore and Hannah Chang. Shannon's an entrepreneur, and Hannah's taken over her family's technology business in California. Nicole Yu, who is now at Johnson & Johnson. Dan Hageman just recently finished his PhD. Lucy No, and Joanna Ung, and Veena Putra. I'd like to recognize their contributions to the work I presented today, as well as the contributions of many funding agencies who made this possible. Thanks so much for your attention and for your patience when everything went black all of a sudden. But now we've got it back online. So I don't know what happened, but my computer but it came back to life.
The joys of technology.
That's right.
Well, thank you so much, Melissa. This was a wonderful, insightful presentation. And I think you hit the nail on the head at the end there with the value proposition and how it's important that not only as scientists and researchers, but as we go into this business realm and what value we're bringing to society with these technologies is spot on. And it's something that I often share with my team at NAI is what value proposition can we express to our audiences and show that we are making a difference and impacting society for the better. And just the sheer fact that you've been able to collaborate with so many individuals and foundations and several students even, I think it's important that the next generation be a part of these conversations, be a part of these research teams. So I commend you for doing just that. And so I would like to open it up for Q&A. So again, if you have a question, please use the Q&A function or the chat function and type in your question and we'll answer it live. So the first question is from Brian Millis, who would like for you to explain more about how the dynamic strain map is created.
Thanks, Brian. That's a great question. So the dynamic strain map is created using a technology called digital image correlation. And you may have seen, I don't know what your mechanics background is, but I think everyone's seen on sports programs where there are reflective dots on players, and the baseball pitcher, the cricket pitcher has this reflective dot on him or her, and someone's videotaping it, and then you watch the movement of those dots to get an idea of how he or she can improve their pitch. We're using that at a smaller length scale to understand the strain patterns on the surface of, for instance, the periosteum. So very similar technology. It's called digital image correlation, DIC. When we first started it, we had to write our own code for it, but now there are programs and commercial systems available. Thanks for the question, Brian.
Great. And then someone asked, what advice would you give the next generation of innovators, particularly as the COVID-19 pandemic has shifted perspectives for now and likely into the future?
Thanks for the question. I think for the next generation, the most important thing is, one, getting lifelong learning. Two, getting outside and observing the world around you, because the world around you is incredibly interesting. Three, don't pigeonhole yourself. Explore as much of the world and as much of education as you can. So I think creative endeavors, you know, I do a lot with artificial intelligence or machine learning, and you can write algorithms to do things that humans do, but I don't think we're going to have an algorithm to be creative or to think creatively and innovatively. So I think exploring the creative arts while also exploring science and technology and informing yourself, understanding maths while also understanding music and art, it's all important. And have fun while you're doing it. And that's not an order. That's the best bit of it, you know?
Right.
Yeah.
I think that advice even resonates with me, even though I'm not a student any longer. It's just to be a life learner is something that I pride myself in having that as a part of my my day-to-day, essentially, of just constantly figuring out what's happening in the world and how can I be a part of some of these conversations, or how can I learn more, be at least more bit informed. So the next question I have here is, you talked about new methods incorporating design and manufacture from the conceptualization of a new material, device, or product. What do you see as the future of manufacturing?
I see the future of manufacturing as crossing these length scales and using, in the past, we've used a lot of what we call top-down understanding to create new manufacturing methods. So you look at a whole system and then you kind of take it apart rather than building it up from the smallest parts. And I think it's the bottom up, the building it up from the smallest parts, that's going to give us the new discoveries, that gives us the new technologies and the new improvements in human life, human products, human health. So in a nutshell, I would say.
Great. And then I think we have time for a couple more questions. And it looks like there's 2 or 3 more. And what role would you say does sustainability, which was a key theme for our conference this year, play in your work? And I think it's something that's important to share across multiple industries as well.
Yeah, you know, I think sustainability it's all about survival. Mm-hmm. And without sustainability, we cannot survive. And that's why I think it's so important. I get so much inspiration from Indigenous people all around the world. Mm-hmm. And indigenous people, they've lived on this earth a long time and have figured out, you know, how to sustain, and that sustains everything around us, everything. So that's an absolute important theme. The other thing is understanding biology is sustainable. Plants don't do things to themselves, so they won't be able to survive.
Right.
So I'll leave it at that.
No, that's a great point. And then lastly, this question here, we have had a number of inventor scholars talk about the promise of nanomaterials, and your work spans length scales from nano to meso. Which length scale is the hardest for you to decipher?
The hardest for me used to be at the smallest length scales. But I would say through imaging now, we're able to see the smallest length scales, and we're also able to much better understand those in context of time scales. And so I think imaging is key, and traversing those length scales is key. And as I showed that multi-hierarchical map, I think that's the way to go. And there are just millions of questions to be answered. So lots to do.
Great. And I'll just end with this comment, actually. So someone wrote, no question, I just want to say your passion in your work is obvious, and they thank you for your contributions and your presentation. And also thank you for your thoughts on the natural world and indigenous people. And so I agree. I thank you.
It's a pleasure.
For this wonderful presentation. And if there are other questions, feel free to email us after this webinar. The webinar will be available for future watch on our YouTube channel. And it was actually live on Facebook today. So if you're wanting to watch it again, you can also view it on our Facebook page. But Melissa, thank you. I know it's late for you. I hope you get some much needed rest, but we appreciate your time today. And it was a lovely presentation. Thank you again.
Thank you, Jade. Thanks to the NAI, and thanks to all of you for watching.
All right.
All the best, everyone. Bye-bye.
Bye.